Well, welcome back everybody to the Cerebral Blood Flow virtual seminar series. Good evening to those in North America. Good morning to those of you in Australia and in Asia. And unfortunately, most of our European colleagues probably can't make it because it's in the wee hours of the morning. But thank you very much everyone for attending today. We're really excited to have our Australian colleagues presenting in addition to a couple of our Canadian colleagues. So thanks so much for joining us. Just a couple of items here in terms of our rules for engagement. These are the same as what we've had in previous sessions, but just to please keep your microphone on off and your video off throughout the session. There will be question time at the end of each presentation. I think the first couple of presentations will go through for both of those and then have questions at the end of the first two. And then there'll also be an opportunity for you to ask questions by either raising your hand in the participants link or posting your question in the chat. So please use either of those two options for questions. As always, this session is being recorded right now. Will be posted on the Carnet website over the next couple of days, so keep an eye out for that. If you would like to be a member of the Cerebral Autoregulation Research Network, please send me your contact info and your CV, and I'll add you to the mailing list. So as I say, we're really excited today. We've got a great session lined up. Thanks so much to the chairs. We've got Bara, Kurt and Ryan again co-chairing this session. So thanks so much to all of you for coordinating this great session today. And Kurt, I'll pass it over to you to introduce Danny Green.
Thanks, Caroline. Ryan and Bara and I started discussing what was the best way to kind of go about taking our second seminar here. And I think between the three of us, we definitely had to agree that Danny was going to be the guy to go to. He's a leading researcher in cardiovascular exercise physiology, and he's one of my primary mentors. He's published more than 300 papers since 1998. He's primarily worked at the University of Western Australia, investigating vascular function, specifically in cardiovascular disease. He's done work in cerebrovascular function, as he's going to discuss today. He's got over $8 million in career funding, and honestly, he's one of the kindest men I've ever met in research. And it's with great honor to introduce him and his team that he's going to present with today. There'll be Danny, and then he'll introduce the remaining talks.
Thanks, Kurt. Thank you very much. That's a very kind introduction, and good morning from Perth. Good evening or good day, wherever you might be. Very good. Right, well, good day, everyone, and thanks for the invitation. I'd like to thank Caroline and Patrice, Kurt, Ryan, and Bara for organising this talk. I did want to start with one slide about how this crazy year is affecting us all, and just put it out there that wherever you happen to be in the world, we hope that everyone's okay. That from us and from our families, we hope that you and your families are safe and stay well in this crazy year that we're all enduring together. A little bit about the Australian response to the COVID crisis. This is March. It seems like the first thing we all did was rush out and buy toilet paper off the shelves, which is a kind of interesting first thing to do, I think. But the Australian response has been actually pretty good in terms of where we're at. This is Australia with obviously the United States planted on it. We're a big country, very large island. Most of Western Europe fits into the size of Australia. It's a country of 23 million people. There are 2.6 million people in the city of Perth. It's a reasonably sized city. And we've had a total of about 700 cases since March. There have been 9 deaths, all in elderly people. All of those cases have either been expats returning to Western Australia or people, there's been no local transmission of the disease since the get-go. We've had an exemplary political response to this crisis, both at federal and provincial level. Right now in Western Australia, there are 16 people with COVID and 2 and 1 in ICU. So we are not locked down. We're face-to-face at universities. There are no restrictions. I went to an Australian rules football game a couple of weeks ago with 30,000 people in the stands. So the fact that we're all sitting here talking to you in one room with no masks on is not because we're Donald Trump supporters. This is a little bit about Perth. It's rated as one of the world's top 10 cities. It's a nice place. It's very isolated. Geographically, our closest other capital city is Jakarta in Indonesia. It's closer to us than either Sydney or Perth. It takes 5 hours in a plane to get, when you can get on a plane, to get to Sydney or to Melbourne from Perth. The University of Western Australia is one of Australia's oldest universities. It's ranked #1 in Australia for life sciences, and the university is in the top 100 in the world by the ARWE ranking system. I put these up to show you because I hope that one day people will be able to travel again, and when you do, if you ever get to come to Australia, you're very welcome in Perth, and it's a lovely place to come and visit when the craziness stops. So this is my job today. It's really to introduce a lot of really smart other people who I have the privilege to work with. So I'm going to be very brief in my remarks and then hand it over to a lot of ECRs to do the heavy lifting in today's symposium. I have got one sort of general platform-like slide. This is the paradigm that we operate on in my groups. And it really just shows you on a timeline which has age on the x-axis. Many of you have seen a slide. It's the sort of time course or natural history of the evolution of atherosclerosis. And I've put in a little section to the right, which is to recognise that our colleagues in medicine do a lot of important, particularly at the moment, with this endothelialopathy affecting us globally. They put in a lot of heroic effort, but it's heroic effort at the end of a process. And there's a long occult phase of development. There's a long gestation period before you get to this end of the spectrum where disease manifests. And it manifests in many ways, and we have lots of ologists to help us when that happens: cardiologists, neurologists, ophthalmologists, nephrologists, vascular surgeons, cardiovascular surgeons. And in Australia, at least, we spend about 10% of our GDP to the right-hand side of that line after disease manifests, atherosclerotic disease. We spend about 2 orders of magnitude less on prevention in the 60 years that it's taken for people to manifest disease of that nature. 5 of the top 10 causes of world mortality, according to the Global Burden of Disease Study, are cardiovascular or atherosclerotic in nature. So there's a massive global burden of these diseases, and they're highly preventable, but we don't spend enough time thinking about prevention or how to prevent them. Just to put things into context, the top 2 causes of global death, which are coronary artery disease and ischemic stroke, cause about 17 million deaths a year globally. I think the COVID thing's up to about over 1 million deaths worldwide now. So infectious disease is making a comeback, but cardiovascular diseases are still the #1 causes of death worldwide, and healthcare costs. So really our paradigm is to do things that will prevent the progression up this way, and maybe even reverse it if that's at all possible, and also to detect disease early. So we really have 2 principal driving themes in my group. One is early disease detection, and the other is optimised prevention. There's no point detecting things early if there's nothing you can do about it, but we're exercise scientists as well, and we know that we can have an effect and maybe even optimise the benefit if we're clever about it. So that's the platform for what we do in my group, and some background for today. Then the other slide I wanted to put up is about early disease detection, and many of you will recognise these now classic studies in physiology which proved that the endothelial cell lining is crucial to vasoreactivity. So in the classic dog hind study from 1986, endothelial denudation, which was generally induced in these experiments using balloon angioplasty or a balloon inflation, endothelial denudation led to a lack of reactivity in response to flow and shear. That's in the dog hind limb. In dog coronary arteries, the study of Bordeaux, which is very elegant, showed that dogs put on a treadmill will have epicardial coronary arteries that will dilate in proportion to the speed on the treadmill, unless you have endothelium removed, in which case you get a dose-dependent or intensity-dependent vasoconstriction, which is not good when you need increased myocardial oxygen delivery. In structural terms, this is a study of Langle and O'Donnell, again from an old study but a classic. I urge the younger people who haven't seen these studies to look them up. They're covered in the physiological review that we wrote a couple of years ago, so that's a starting point. But go back to the source articles. They're very elegant, beautiful science. And this is a study where one rabbit carotid artery was ligated, and when you have endothelial cells, you actively remodel that vessel structurally, but if there's no endothelium, all of that structural remodeling goes away when you decrease the shear. So all of these studies prove that shear stress is a really important stimulus to function acutely of the arteries, but also to longer-term structural remodeling. And in studies done by Ellen Dawson in Liverpool, she's really reiterated most of this data in humans using a radial artery denudation model, secondary to catheterization. So in humans and in lots of different animal models, you see this. So the summary is that flow and shear stress activate the endothelium, and they're a physiological stimulus that does that. Just in case you're interested in shear stress per se, here's some computational fluid dynamics. You'll be hearing a lot more about this in a minute from Harrison, who will give us an engineering perspective on how this is done. But here are some flow vectors that have been calculated from our duplex ultrasound inputs and MRI for the structure of the brachial artery and its branches. And you can see that there's a change in shear stress across the cardiac cycle. But when we institute leg exercise on a treadmill, this is a resting brachial scan, all hell breaks loose. And there's all sorts of reverse jets during diastole. There's lots of interesting things going on with the shear patterns here. So more about computational fluid dynamics in the context of the cerebrovasculature coming up. In a series of studies I'm going to summarize very briefly, I think we really showed quite elegantly that shear is important to vascular adaptation chronically. So this is really a summary by a lot of really hardworking people over here, showing a series of studies we did with different interventions. These are all longitudinal experiments, internal controls where we studied the brachial artery in one limb, and in the other limb of the same individual, we had a cuff inflated during each of the sessions that we undertook, and that was bilateral hand grip exercise training over 8 weeks, or leg exercise training where we studied the upper limbs, or without exercise, just bilateral forearm heating. And the point of putting this cuff in series here is that the brachial artery is upstream, and inflating a cuff just to 60 millimeters of mercury decreases the flow and shear stress in this vessel during each of these bouts of exercise. So you've got a very elegant within subject bilateral control here, where we've clamped shear stress but exposed both limbs to the same stimuli. So this is a way of dissociating shear stress from the exercise or heating stimuli. And to summarize these studies very briefly, we see a change in function in the artery. This is endothelium dependent, largely nitric oxide mediated function, FMD assessment. But in the other limb, which got exactly the same stimulus but without the shear, no change whatsoever. And there's some evidence for structural remodeling in these conduit arteries after a period of time, which may compensate for a sort of peaking over of the functional response. So there's an interaction between function and structure, but again, no structural adaptation. So these chronic studies really do reinforce the animal data I showed in the previous slide, and also reinforce the importance of shear stress to vascular adaptation, either chronically and acutely. Why is that important? Because we don't really understand how exercise works fully. And to explain that, Mike Joyner helped me a couple of years ago to put a bumper sticker on this and called it the risk factor gap. And we've written about this for many years now. Basically, it says that the fairly large and impressive impact of exercise training or physical activity on all-cause or cardiovascular mortality, that's a 30 or 40% benefit in most of the epidemiology, cannot be fully explained by the effects of exercise on risk factors. In fact, if you add all the risk factor modification benefits up, they only account for about 35% of the heart disease reduction. So there's a lot of unexplained benefit of exercise, which is a good news story, because people who exercise, train and don't change their blood pressure or their lipids very much are probably still getting a benefit. And we would argue that that benefit is largely mediated by hemodynamic changes, which are having a direct, perhaps biomechanical effect inside the vessels themselves via shear stress. So our peripheral artery platform, the filter we bring to this work on the cerebrovasculature, is summarised as follows. Endothelial dysfunction is an early and integral atherogenic event in humans. Shear stress is the principal physiological stimulus that enhances endothelial function and health in humans. Exercise increases shear stress incidentally, and for another study, so does body heating, local heating. Repeated episodic exercise or exercise training, physical activity, upregulate endothelial cell function. We really know this from the profoundly important legacy of Harold Laughlin and his group, as well as others. So repeated training improves function, and that also leads to arterial remodeling and enhances vascular health systemically, not just locally in the active muscle. And finally, exercise mediates some of its cardiovascular effects by direct arterial effects rather than indirect effects mediated secondarily through risk factor modification. So that's all fine, and that's what we brought to the table about 10 years ago when I teamed up with this character on the left. This is Phil Ainsley, and I think this captures him either wistfully looking into the academic middle distance and posing the question, what about the cerebrovasculature? Or he's just had one too many exposures to hypoxia, I'm not quite sure. But why should we be interested in this idea about shear stress in the cerebrovasculature? Two very brief slides which can be expanded on in question time if you like. This one recent from the Whitehall study, 3,000 participants, mostly white, mostly male, English civil servants, studied between 2003 and 2005 when a carotid ultrasound was performed among all of the other myriad measures they took of risk factor profiling, and studied again 10 years later. And in this study, they showed a very strong independent effect of the carotid forward compression wave intensity. So this is the beginning of the systolic waveform in the carotid artery, and it's strongly associated with cognitive decline. And it's in fact the most strongly correlated factor that they were able to find. And that's independent of other cardiovascular risk factors that have been linked to cognitive decline. And the conclusion from this study was that elevated carotid artery wave intensity, the hemodynamics in the carotid artery, in mid to late life predict faster cognitive decline and longer-term follow-up. And that's independent of other cardiovascular risk factors. So some clear link here between some factors that control blood flow and perhaps then shear stress in the cerebrovascular tree and cognition. There's also reason to believe in this cartoon that we generated that there's some links between angiogenesis and neurogenesis in the central nervous system. And this evidence is indirect, but there's quite a lot of different lines of evidence, mostly animal-based. But there's clear links between cerebrovascular dysfunction and Alzheimer's. There's links between endothelial dysfunction, which is associated with dementia. Amyloid induces endothelial dysfunction. Nitric oxide, endothelium-derived nitric oxide production protects against increases in amyloid in animal models, whether you believe the amyloid hypothesis or not. So this led us to propose that perhaps there's something going on in this space between the angiogenic factors that are at play in the central nervous system and neuronal generation, and perhaps finding some way to improve nitric oxide production could help could go some way to explaining the beneficial impacts of physical activity and exercise on cognition with age. So this leads me to this slide, which is really dedicated to the relationship I've had with Phil over the years. Phil has been a terrific collaborator. I think we've set up a really amazing collaboration given the distances between the groups. We've had lots of people visiting between. I've visited UBC 3 times, most recently in March this year. I had to get out quickly. Phil's supposed to be here again this year. That will make 4 visits in the last decade. Kurt spent 3 years with us as a postdoc and did a lot of great work. Ryan's been over here for a while. We had Chris Willey with us for a while, Daria, Alex, lots of visitors, and from my group, the other direction. So this is really an exemplary example of how collaboration can work internationally when it's based on mutual respect. And I think that's the great thing about Phil and I. We have a large amount of mutual respect for one another. And it's been a great collaboration over the years, and I credit Phil publicly for that. So this is really a cerebrovascular speed dating event. I've said my bit. I'm going to flip it straight to Howard now. He's going to continue on the theme of how we can manipulate cerebrovascular shear stress acutely and then chronically and see what happens in terms of cerebrovascular function measures. Hannah and Harris are going to talk about quite specifically about some ways that we're trying to improve the techniques that we use to assess cerebrovascular shear stress. And then Kurt's going to talk about some really unique work he did while he was here in patients with end-stage heart failure and ventricular assist devices implanted. And then we'll have the UBC crew come in and talk to us about some aspects of the work that Phil has been continuing in collaboration over there. So without further ado, I think I'll flip it to, sorry, this is my summary slide. I think my sort of executive summary would be that we still need better tools to measure the cerebrovasculature. We're trying to do that using some computational approaches, but there are limitations to all the techniques that we use at the moment, and there's still a lot of work to be done in that space. Those tools really need to be able to detect early dysfunction, because if we're really going to have an impact as a group, we need to detect the disease early before it's clinically manifested and largely irreversible in terms of its impact. We need clever randomized controlled trials that try and isolate the effects of exercise, and even more specifically, the effects of exercise on shear stress and hemodynamics. And Howard will talk to you about how we've made an attempt to do something like that. So that's me. Over to Howard Carter, who's going to talk about the Preventia study.
Yeah, thanks, Danny. So I'll spend the next 10 minutes talking about a series of studies we've done on water immersion over the last couple of years. So when we entered this sort of research agenda, there's obviously been a long history of research into water immersion and human physiology, and this started back with respiratory mechanics around looking at the pressure-volume relationship, and their studies as early as the 1900s, probably earlier. And then in the 1960s, there was a lot of research done in the 60s, 70s, 80s with the introduction of space exploration and the interest in microgravity. So this is just an image of the Apollo astronauts practicing for their space flights in the neutral buoyancy simulator in America. But around this time, we found out a lot about the effects of water immersion and hydrostatic pressure. And we knew that water immersion in someone standing upright centralizes blood volume. You get an increase in venous return. This stretches the heart, increases preload, which improves stroke volume, and in general increases cardiac output. But what we found interesting and I guess surprising was that there was relatively little known about the impacts of water immersion and cerebral blood flow. And this is particularly true for water temperatures between 30 and 35 degrees. So that's what led us to our first sort of proof of principle study, and that was to determine the impact of water immersion in 30 degrees on cardio, but specifically cerebrovascular variables. So it's quite a simple but elegant study designed this. So we instrumented the participants with TCD. We looked at respiratory variables and also systemic hemodynamics. And this was collected at standing and then during filling and for 10 minutes. And then we looked for reversing the signal by emptying the tank as well. So that's possibility. But so we found, consistent with the previous literature, a significant impact of water immersion on mean arterial pressure. We found increase in stroke volume. significant changes in heart rate and an increase in cardiac output. But more interestingly for us, we found a significant impact on middle and posterior cerebral artery velocities with immersion, as you can see here, in the range of 10–15% increase. So this is quite significant, particularly in terms of the brain, which is so tightly regulated. So the next question for us was what happens during exercise. And so a follow-up study, Chris Pugh here, performed a study in water, so it's the same temperature, and on land, in 15 healthy subjects. So they performed 20 minutes of stepping exercise in water, and then on land, and that was a randomized approach. And you can see here the water and land exercise bouts, they were matched for intensity. So you can see VO2 here was the same, as well as heart rate. So that really allowed us to compare the cerebral blood flow responses nicely. And the difference in heart rate at rest, you can see that's consistent with our earlier study. So this was looking positive. And you can see here just the systematic impact of water immersion on middle and posterior cerebral artery velocity. So the bottom line's the land exercise and the top line's the water exercise here. So that's a really significant difference, and it's maintained in a very similar pattern between land and water exercise. Again, in the range of 10–15%. So we've shown this, and it's been great to see people like Kiwi mate Sam Lucas, Jim Cotter, Kate Thomas, Lena Shoemaker, and Blair Johnson as well. I think they've all shown a similar impact of water immersion on cerebral blood flow, either at rest or during exercise. So it's quite definitive, the impact of water immersion. And the question is, so what? So we've shown that water-based exercise induces a greater increase in cerebral blood flow. So the importance of this comes, it's in the context of the work that was done on peripheral vasculature, so work by Danny and others, that showed the impact of exercise on blood flow and shear stress, and how that improves endothelial function here, improves vascular health, and ultimately can reduce cardiovascular risk. So the idea was to take this concept from the peripheral vasculature to the cerebral vasculature. And the question being, could recurrent increases in cerebral blood flow improve cerebrovascular health? And this is what led to the Preventia study. So this is the first large collaborative trial between Danny and Phil, and also some neuropsychologists and psychologists. So this was a clinical trial run at EWA for over a number of years. It was a big undertaking. So as I mentioned in the previous slide, the rationale for Preventia really was, we know exercise is good. It improves vascular elements of brain health. It's effective at primary, secondary prevention of cerebrovascular disease, but we don't exactly know how. If we think it's similar to the peripheral vasculature, and we think it's blood flow and shear stress, how do we test it? That's where the water immersion intervention comes in. So if we recruit individuals to undertake a 6-month exercise intervention in water, where you get this response, as opposed to a group who undertake land exercise for 6 months. The first question is, would you see cerebrovascular adaptation in either group? But more importantly, because of this augmented cerebral blood flow response during exercise in water, would you see a greater adaptation response in this group here? So that was the rationale behind Preventia, and that's the results that we'll talk today. So the participants were subjective memory complainers, so just older individuals who subjectively had issues around their short-term memory, but this was in the absence of depression and mild cognitive impairment or dementia. They were 50 years or older, and they were healthy but otherwise inactive. We had a lot of interest. It was a very tightly controlled study, so we had over 900 people contact us, and through eligibility criteria and withdrawals, we ended up with 72 people enrolled in this study. So it's 21 in the control group, 23 in the land, and 21 in the water intervention group. So these individuals had the cerebrovascular assessments and then continued on for a 6-month training intervention. So it was a supervised center-based exercise, 3 sessions per week. The land-based walking performed their exercise on the university grounds and in the surrounding parklands. So this is next to Swan River. It's a beautiful place. And the water walking group was, they did their exercise in the 28 heated pools at the university campus. This just shows the exercise intensity was matched between the groups. throughout the whole 6-month intervention, and you can see the intensity was ramped from week 0 to 24. So the exercise physiologist did a really nice job there. In terms of methods for cerebrovascular function, we used transcranial Doppler, and Big Kurt Smith had a significant role in this study. And so we looked at neurovascular coupling, so that's looking at the posterior cerebral artery response to visual stimuli. We looked at dynamic cerebral autoregulation, that was using a lower body negative pressure machine, using 10 seconds on off cycle. And we also looked at cerebrovascular reactivities, looking at the middle cerebral artery response to 3% carbon dioxide and also 6% carbon dioxide. As for the results, these are resting cerebrovascular variables, so control group, pre and post. So no change in middle cerebral artery velocity or conductance, posterior conductance. That's true for the land walking exercise as well. And also we found no additive effect of the water walking in terms of resting cerebral perfusion. For our functional assessments, the neurovascular coupling, you can see a non-significant decrease in the posterior cerebral artery change with the visual stimulus. So no change in control, no change in land, and no change in water walking group. For our CO2 reactivity, this is velocity reactivity slopes. There was no impact of the exercise intervention, either group or in control. You can see water walking decreases slightly here, and what we found was with the MCA conductance reactivity slopes, a significant difference between the control and the water walking group, and you can see a bit of a decrease there in the water walking group as well. We also pulled the exercise data, groups data, and we looked at the change in VO2 fitness and change in reactivity slopes, and we found that there was a strong negative correlation. So that all that says is that in general, for individuals who improved most in their fitness, they generally had a lower or reduction in the reactivity slopes. And again, so the suggestion being that perhaps exercise has a beneficial impact by reducing the reactivity slopes. And this fits in with the concept that some studies have shown reactivity to increase with aging in humans. So definitely something that's worth following up on. And finally, the autoregulation. No difference in gain. You can see a slight decrease in the water walking group. No difference in phase or coherence, but a significant difference between the exercise groups for normalized gain, with a decrease in the water walking group, which would be interpreted as a beneficial change. So, concluding comments for this study is that we used the novel application of water immersion to manipulate the cerebral blood flow during exercise training. Exercise training can augment some aspects of cerebrovascular function, but not all in an older population. And the findings in the water-based exercise group and the signals we saw there does suggest that the hemodynamic stimulus during exercise may be a key mediator of adaptation and potentially similar to what's seen in the peripheral vasculature. So we find water immersion research really interesting. We think it's got a good future, and it's good to see other groups also performing work, and we're also doing some follow-up studies looking at the effects of water immersion on cerebral blood flow. So that's the end of my talk, and I think it was question time. Yeah, I think. So happy to. Yeah, over to Kurt. Do you want to?
Thanks, Howard. There's no questions right now in the chat box, but people might typically warm up a bit. I've got one question for you though, Howie. I've always been curious about the impact of the water on the cerebrovascular response to exercise. Do you think it's a product of posture? Would we see the same if they're in a supine position versus an upright position?
Yeah, absolutely. I think this is Luna Shoemakers and Kate Thomas and Jim Cotter. So they looked at that, and they found that if you're supine in water, there's no difference in cerebral blood flow. So absolutely, it's a postural thing. It's a gravity thing. So if you're lying flat, there's no real gravity impact. So you have to be supine for there to be an impact on at least cerebral blood flow. And it is interesting, I think, yeah, like I said, Blair Johnson's group, they've looked at it and they've tried to work out what it is with water immersion that increases cerebral blood flow. We see an increase in blood pressure. That was in the first study, but we also saw an increase in PET CO2. But Blair Johnson's group, I think they've replicated the increase in CO2 that you see with water immersion in the absence of water immersion, and showed that that doesn't account for the increase in water immersion. So if that makes sense, it's not the impact of CO2. So it just seems to be, it's an integrative response of water immersion. So the impact on cardiac output, blood pressure, PET CO2, it seems to be a grouped effect that just, increases cerebral blood flow without stimulating any sort of reflex auto-regulatory features. So,
awesome.
That argue that it's stroke volume related.
Great. I've got a couple, as I said, I knew it would warm up a bit. So Tracy Baynard has a question. Given that one of the variables was impacted by training within the water immersion group, do you feel there are more sensitive measures that might yield something of interest if you could measure anything you wanted?
Yeah.
Hi, Tracy.
Yeah. I mean, as everyone probably knows, we're limited with how we can assess cerebrovascular function. You would kind of throw the kitchen sink at it when we look at neurovascular coupling, autoregulation, CO2 activity. But in terms of sensitivity, I definitely think that more sensitive tests might uncover some changes. And if we're just looking at resting cerebral perfusion, that seems to be a very important variable in terms of cerebrovascular risk. And we saw no impact of the exercise in either group on resting perfusion. But if you look at the animal studies, if I'm allowed to do that, it generally shows that with exercise training and fitness, there's no impact on total cerebral blood flow, but there's regional impacts of blood flow on perfusion. And there is some evidence in humans, so there's at least 3 studies in humans that have looked at exercise with MR, and not showing any impact on total cerebral perfusion, but showing regional impact. So I think with better imaging technologies, rather than looking at global MCA and PCA flows, you might be able to have to look at more,
Can I comment on that?
Yeah, please.
So firstly, hi Tracy, and thanks for your question. I hope everything's okay over there. 2 things. One is, it's going to be difficult to mount an MRI machine above our water tank anytime soon. So there are limitations to the technologies we use. The other thing I like using the word sanguine for obvious reasons about these blood flow effects in the brain, because I don't think the shear stress amplitudes we see, they're more highly regulated than they are in the peripheral vasculature. So, with skeletal muscle exercise, you're seeing 10-fold, 20-fold, 30-fold increases in blood flow. And you saw the shear stress image I showed earlier from the CFD we did. It's radical and impactful, maybe not so much. And so the stimulus, or the magnitude of stimulus, is going to be less. The signal is going to be harder to detect. So we've got 2 problems, probably a small signal and difficulties in detection. And, maybe we'll see a resolution of that by finding a way to manipulate the brain blood flow a bit more than we did. It was, 15 or 20% based on the water immersion. I would have thought that would have been close to enough chronically to see a benefit, but the benefits we are seeing, if any, are subtle.
Great. There's a couple more questions here. We're going to ask Mike Timko, living in Edmonton, hopefully staying dethawed. He asks, what was the relative cerebral blood flow response to exercise in both the MCA and PCA the same between land and water immersion?
The relative change was very similar, yeah. The relative change from rest to exercise.
The increase from rest.
The increase from rest was very, very similar. We can, we can go back to that slide, but yeah, in terms of a pattern response, it was almost identical.
You mean rest in the water though, not rest that day. You know, so the magnitude of increase when you're immersed is higher compared to what you're doing for the rest of the day, which is matched. Yeah, just I think that's an important point. Sorry.
Yeah, I'm not sure if I answered that.
No, I think you got it.
That's good. We'll take one more question from Caroline. Great talk, Howie. Were there any differences in the arterial pressure responses to the cerebral test following the exercise intervention?
Which cerebral tests?
Yeah, we did 3 sets of cerebral. Yeah, I guess. We did.
Sorry, Kurt. Which cerebral test?
I imagine either this. I imagine the lower body negative pressure, or I mean, it could also be the CO2 test. I guess if blood pressure was significantly altered pre and post.
Yeah. So the question was the blood pressure response to potentially CO2 activity different,
or LBMP different between pre and post interventions? Yeah.
Yeah. So I think with the conductant CO2 activity data, with the change we saw there, it did seem like potentially there was a lower pressure response to CO2 activity post intervention in these guys, which I think accounts for the change in CO2 activity. In terms of LBMP, with the way that that's analysed, we didn't look at the absolute changes in mean arterial pressure, so I can't really comment on that.
But we published data on the blood pressure, I think, from Preventia, and I don't think there was a strong signal around resting. We measured blood pressure after 30 minutes of quiet rest using Dynamap, in an isolated room, etc. We were interested in blood pressure effects per se because of the hemodynamic effects of water. And my recollection from that data, which I think was published in one of the Haynes studies, was that we didn't see a strong signal for blood pressure change in the experiment per se. So I think that goes some way to answering the question.
Yeah, and nor do we find it in the blood pressure from the cerebral.
Yeah, that answers
results.
Great, guys. Howard, great talk. There actually are 2 more comments or questions in the chat. I don't know if you guys feel like trying to find a way to respond to any of them. Just as we go, and what we might be able to do is move on to Hannah and Harrison.
Okay, so that was a really nice start to our seminar. Thank you very much, Danny and Howie. I don't know if anyone else felt this, but it felt like we were getting an insider view to what it's like to be at one of Danny's lab meetings. It sounds like it's a fun group to be a part of. Our next speakers are the dynamic duo from University of Western Australia, Hannah Thomas and Harrison Caddy. I first saw their work on Twitter, and I thought it was a wonderful display of the cool and innovative science that can be done when interdisciplinary forces come together. So without further ado, it's my pleasure to present to you Hannah and Harrison, who will share their work on the assessment of cerebrovascular function in twins using multimodal imaging and computational fluid dynamics.
Fantastic, thanks, Bara. So as Danny mentioned earlier, my background is more in computational fluid dynamics, or CFD, as I'll sort of refer to it. So first, I just wanted to kind of give everyone a bit of an overview of how we've combined some modalities into CFD, and then Hannah's going to explain how she was able to apply this in an experimental context to twins. So first, just touching on some of the different ways we can measure cerebral blood flow, and I'm sure everyone is well versed in these, but we have duplex ultrasound, which is great for obtaining a flow measurement for blood leading to the brain, so long as we can get a velocity waveform and a diameter envelope, which is some software that Danny's group has really pioneered over the last decade or fair while. But this is limited to the neck artery, so it's great for really understanding flow to the brain, but not really flow within the brain. We have transcranial Doppler, which is great for getting the velocity, but unfortunately we can't really get diameter measurements and therefore don't really understand the flow there. It's also somewhat dependent on how the user places that insonation region. And again, due to the shape of the brain, we're limited to the vessels that we can insonate. Now there's forwarding flow MRI, which is fantastic. However, again, you're kind of limited to the conditions that you can put someone within an MRI machine. And unfortunately, the temporal resolution just isn't really the same as that of ultrasound. So what can we do about this? We can start looking to combining modalities. So we can try and use that duplex ultrasound to give us those flow waveforms, which has great temporal resolution. And if we can combine that with the magnetic resonance imaging, which has great spatial resolution of blood vessels within the brain, into a computational fluid dynamics simulation, this might enable us to capture really accurate measures of shear stress within the brain. So the computational fluid dynamics method, or CFD method, really revolves around 4 key processes. So first, we need to obtain a 3-dimensional geometry. So this is done through image segmentation to get a 3D surface. We then need to fill that 3-dimensional surface with little control volumes, and that's in the volume meshing step. And that's so we can solve these equations called the Navier-Stokes equations, which is kind of solving for the convective and advective terms of fluid transport. We then need to sort of prescribe physics models to those, to the fluid boundary, as well as boundary conditions as well. And then we have to, of course, simulate and extract our results. So first, touching on the segmentation, we took our DICOM images, we reconstructed 3-dimensional models in in-house software that we have here. Here we also performed some global smoothing and dilation changes with a volume conserving metric. I think it was around about 5% conserved. And this is great for getting most of the vessels. However, as you can see here, there's still some artifacts from the voxels being too close together, and this isn't considered physiologically accurate. So we needed to perform some CAD operations or computer-aided design operations where we essentially unwebbed these webbed vessels where they came up, and we also performed a coarse truncation. And then from there, we're able to fit a center line and cut the outlets perpendicular, which is quite important for keeping the outlets to your fluid simulation stable. So from there, we needed to fill this 3-dimensional surface with a whole bunch of little volume elements, and we use a combination of polyhedral cells for the core of the mesh. As you can see here, this is a cross-section of a vessel. And then at the wall, where we have high shear gradients, we use a layer of prismatic layer element cells, which is able to capture that high shear gradient. So our simulations ended up with around about 16 million cells per case. And we performed a discretization error study on this, and this was found to be below 3%. So we deemed this to be adequate for our simulations. So here you can see we've taken that volume meshed, this is an example of a volume mesh geometry, and we've imported it into a commercial CFD solving program. This is the same kind of program that we use to understand fluid flow over a car, for example. We then prescribed laminar flow for the blood and assumed that it had a non-Newtonian viscosity. And we used a rigid wall boundary assumption at the wall, and also assumed that the fluid couldn't, no slip conditions, the fluid traveled at the same speed as the wall. For element boundaries, we used the ultrasound flow measurements at the neck leading into the cerebrovascular. And at the outlets, we didn't really know where the flow is going to. We based our flow splits within 7 different regions within the brain, and we wrote a little bit of program called Walnut. And if anyone's wondering why it's called Walnut, it's because walnuts look like brains. So once we've done that, we needed to execute our simulation. We ran our simulations for about 3 cardiac cycles, and we extracted data on the 3rd cardiac cycle. These simulations are quite computationally expensive, so fortunately here in Western Australia, we have access to the Palsy Supercomputing Centre, particularly this computer called Magnus, which is this 35,000 core supercomputer, which is a fantastic resource. And each simulation required around about 2,000 core hours per case, per condition, as Hannah will explain in a second, to run. So here's an example of some of the sort of shear stress and velocity information throughout a cardiac cycle that we can extract. And this is a nice video and all, but really we're kind of more interested in how we can extract measured data in certain regions. So we looked at time averaged wall shear stress variables within the circle of Willis. So the first metric here, TAWSS, stands for time average wall shear stress. We looked at something called oscillatory shear index, which is kind of the oscillatory nature of the shear vector at the wall, and that's something that Danny talked about in those exercise videos that he played earlier. We also have this metric here called relative residence time, or RRT, and this is a measure of the relative time that blood might reside near the wall. So I'm now going to pass over to Hannah, who's going to explain how we actually applied this in a twins context.
Great, thanks, Harrison. So I'm just going to, as Harrison just said, talk a little bit more specifically about the study that we did using this technique. So firstly, the overall aim of the study was to develop a platform that enables characterization of the structure and function of the cerebrovasculature in twins. So firstly, we wanted to combine duplex ultrasound and MRI to visualize the cerebrovascular anatomy and measure blood flow and hemodynamics. We then wanted to calculate shear stress and flow drive metrics to understand the link between structure and function using CFD. And then lastly, we wanted to study the behavior of these arteries under different physiological conditions, so rest, hypercapnia, and exercise. The null hypothesis of the study was that both the structure and function of the circle of Willis, internal carotid arteries, and vertebral arteries would not differ between the monozygotic twins. So for this pilot study, participants had 2 testing visits. Visit 1 involved a head and neck MRI, which enabled us to determine the structure of the circle of Willis. And then on visit 2, we conducted functional assessments using ultrasound. So we used transcranial Doppler ultrasound to measure cerebral blood flow velocity in the middle and posterior cerebral arteries. And then we also used duplex ultrasound to measure both the velocity and diameter of the internal carotid and vertebral arteries. And then we used our wall tracking software to give us measures of total cerebral blood flow through the 4 inlet vessels. So we then combined these outcomes using CFD, giving us the measures of shear stress, which Harrison explained. And then these measures were calculated for the 3 different physiological conditions: rest, hypercapnia, and exercise. And in this study, we recruited 6 pairs of monozygotic twins. We had 3 male pairs and 3 female pairs. And we also recruited 6 pairs of dizygotic twins, but for today, I'm just going to speak about the monozygotic data. So the results of this study involve a number of large tables. So I'm just going to summarize each of the main points. If anyone's interested in looking at the data a little more closely, feel free to jump onto JIP, as this paper was just published recently. So this figure is showing us the 3D MRI reconstructions of each participant's circle of Willis. So there are some obvious similarities within the twin pairs, and we can see that there are a lot of noticeable differences in the structure. So for example, if we have a look at case 11 and 12, so these are a twin pair, and we can see here that case 12 has an incomplete left posterior communicating artery, where their twin case 11 has a fully intact left posterior communicating artery. So before we even look into the specific measures of structure and function, we can see that there are already marked differences between the twin pairs. All right, so first we have the results for structure. So we calculated the intra-class correlations and their respective p-values for vessel diameter, length, tortuosity, and bifurcation angles of the 4 of the circle of Willis segments. So a high intra-class correlation would indicate that the twins within a pair are similar, and a low correlation would indicate that they're different. So we found that there were only low to moderate correlations of the circle of Willis structure between the monozygotic twins. And this would indicate that the structure of the circle of Willis is highly impacted by both shared and external environmental factors, with minimal influence from our genetics. So next we wanted to have a look at the functional measures, so both at rest and in response to the hypercapnia and exercise stimulus. So firstly, both diameter and flow in the internal carotid and vertebral arteries presented only low correlations between the twins. And secondly, the shear stress variables again showed only low correlations between the twins, which indicates that similar to structure, function at rest and in response to the physiological stimuli are highly impacted by our environment. So to just bring it all together, our aims were to investigate the variance seen in the circle of Willis geometry and understand the influence this has on cerebral blood flow and vessel function under different physiological conditions. And what we found was that despite similar physical characteristics and identical DNA between the twins, vessel structure and function differed greatly. And in addition, the cerebral arteries behave surprisingly differently between twins in a pair in response to the physiological stimuli. So the results of this study are consistent with previous research. Bianca Forgo and colleagues in 2018 were the first to investigate circle of Willis variants with MRI using twins, and they concluded that there were marked differences in the circle of Willis structure and that these differences were determined by environmental rather than genetic factors. So our study added to this literature, firstly by using 3 Tesla MRI for all participants. So before we began the study, we did sort of like a little pilot. We did a 1.5 Tesla and a 3 Tesla scan on one pair of twins. And after analysis, we found that the 1.5 T wasn't sufficient to accurately segment all the circle of Willis regions. So I think it's really important for future that we move forward with high resolution MRI. Oh, sorry. Secondly, we added duplex ultrasound and computational fluid dynamics to provide measures of function. So this allowed us to compare the twin circle of Willis in much more detail compared to simply measuring the region variations. And lastly, we use DNA tests on our twins. Most studies utilize self-reporting questionnaires, but we found that almost 25% of the twins who came into our larger training study self-reported their zygosity incorrectly. So DNA testing is very important. Future research should recruit a large sample size and potentially study a greater age range of participants. And lastly, investigating the changes in circle of Willis structure and function in response to exercise training could provide insight into what factors may affect the circle of Willis changes. Our lab has just completed a large training study on twins. We're hoping to publish the cerebrovascular data very soon. So thanks to all our supervisors and of course all the twins that were involved in the studies.
That was awesome. Thank you guys. Okay, so we'll open the floor to any questions. We have one question right now from Tracy Bernard. She said, I'm sorry I missed it, but how old were these twins? And in essence, were they living together, experiencing somewhat similar environmental and possibly physical factors? And she says, it's fascinating the differences not only in structure, which may be surprising, but also differences in function.
Yeah, so sorry, I didn't mention the twins' ages, but for this particular sub-study, the twins were between 18 and 30 years old with an average of 25 years. There's a range of differences between like how long the twins had been living together. Most of the ones in this study were still living together, so they definitely would have experienced a large amount of like shared environmental factors. And similarly, they did have like pretty similar weights and those sorts of things. So I'd say that they were quite similar in their lifestyles, but it'd definitely be interesting to look at what factors does influence the differences in their cerebrovascular function and structure. So potentially in the future, we could look at their blood pressures or their weights or their heights and see if that correlated with them being more different.
Maybe mention the benefit of including diazygotics for the calculations.
So when we move forward and we look at the diazygotic data, we'll be able to do statistics that enable us to get actual proportions of genetics versus shared environment and then external environment. So we'll be able to say more conclusively what area is contributing to the differences.
Okay, awesome, great. And then Carolyn Kaufman says, really interesting study. Do you think these differences could be manifested at birth, and or does the circle of Willis structure and function grow increasingly disparate throughout childhood and adulthood?
So yeah, really good question. I think that just reading the literature, the main variance in the circle of Willis, so for example, the image that I showed where one twin had a missing LPCOM, I think that those differences would occur before birth, during like the formation of the circle of Willis. But I think over the lifetime, the differences in like hemodynamic stresses could potentially cause more differences, but definitely the segments, like having a whole missing segment, would occur in utero.
Okay. So wait,
yeah, sorry, Danny just wanted to comment.
I think that was, Caroline, that was a great question, and it's one that's taxing us here. One of the slides I put up early in my talk was the experiment where rabbit carotid arteries were ligated over a fairly short period of time, I think it was 6 weeks. And the size of those arteries halved in 6 weeks. So there's clearly demonstrable impacts, and that was a carotid artery, on the size of these arteries. It's quite dynamic and plastic, the vasculature, at least it is peripherally. But Hannah's right. I think that something that's just missing has got to be intrauterine or developmental and possibly epigenetic effects. So it's fascinating that we didn't see higher correlations in monozygotic twin pairs who were genotyped. Yeah, thanks for your question.
Okay, and I have a small question too, if that's okay. You were talking about if what it would look like if you had increased resolution. So to stick with the higher resolution stuff, do you think that there's a balance between how computationally intensive these CFD stimulations are versus wanting to have higher resolution to have more, you know, outlets or boundary conditions? Like, is it worth going even higher to say 7T, or is that going to take too long to run a simulation like that?
From the simulation side of things, evidently the more complex you make it, it generally does get more computationally intensive. But I don't think it really scales. It doesn't get, you know, if you double the resolution, it doesn't necessarily take double as long. I don't think it scales that way. But within our lab, we definitely do test how many bifurcations we keep, say, for example, downstream of the circle of Willis. So that was something that we've sort of tested over the last period of, you know, a while, and we find that as long as you capture I think we found maybe 2 or 3 bifurcations downstream of the circle of Willis, that even if you maybe have different outlet boundary conditions for bifurcations downstream, it doesn't actually affect the measurements within the circle of Willis at that point. So we kind of see it as a bit of a diminishing returns after, you know, a certain period. But
3 Tesla's about right.
Yeah, I think as Hannah mentioned, the real importance is being able to segment out the actual vessels if they're able to be visualized. So for example, if you can't see your communicating artery in a 1.5 Tesla, then that obviously affects the entire simulation. So yeah, I think at least 3 Tesla, but I mean, it would be interesting to see what 7 Tesla would be like.
Awesome. Well, thank you so much, you guys. I will pass this on over to Ryan to take care of the rest of the seminar.
All right. Hi, everyone. So up next, we have Kurt Smith. While he gets his slides ready, I'll provide him an introduction and hopefully change your minds about him after seeing that last photo. But Kurt Smith is an assistant professor at the University of Illinois, Chicago. I was fortunate enough to meet Kurt when I was an undergrad at UBCO while he was doing his PhD with Phil Ainsley. And from that point on, in 2012, I believe, Kurt has really been like a big brother to me, so I'm super happy to introduce him today. Kurt then went on to complete a postdoctoral fellowship with Danny Green at UWA, as you heard, and that's where he conducted the research that he'll present today. I, again, was lucky enough to go work with Kurt and Danny for 6 months in Australia. It was a phenomenal experience, and so when Danny puts up those photos of Perth in his talks and welcomes you to come, I definitely would suggest that you try to take advantage of that. It's a great place to go from a lifestyle perspective and a great place to go from an academic perspective where you get exposed to some really interesting ideas. And so without further ado, Kurt will be presenting on cerebrovascular function in left ventricular assist device patients after an exercise rehabilitation program.
Thanks, Ryan. And thanks, Danny and crew, for those pictures. I forgot how good I looked with the 3-year Australian tan. It's been a really interesting journey working with both Phil and Danny over the years. And there's been some great talks already from Danny's group, and there's going to be some good talks from Phil's group coming up. So I'm going to just kind the progress of, my talk here really is the progress or the expansion of my PhD thesis with Phil, where we were focusing on cerebral blood flow during exercise. And there's been some great talks throughout the seminars where we've learned to define cerebral blood flow during exercise in different forms. So I want to be specific in this talk. This is really just looking at the biphasic response of incremental exercise. The picture I'm about to show here is really just a legendary photo or a figure from Phil and Danny Green's lab. I presented the percent change in cerebral blood flow during incremental exercise in a journal club that Danny was present in 2013. And I tried to rapidly talk through the biphasic brain blood flow response. Danny then proceeded to point out and ask questions about each one of these studies. And I had a kind of a 10 to 15 minute prepared talk. And we ended up talking about this slide for 2 hours. So given the fact that I'm about to switch from that photo, I've already improved from the last time I tried to present this data. Brain blood flow during exercise is largely regulated by CO2, and we see that here when we look at the temporal response in brain blood flow as we go from rest just to maximal exercise. So submaximally, we have a vasodilation that we believe is related to hypercapnia, and then a reduction in brain blood flow that we believe is somewhat caused or primarily caused to a hypocapnic vasoconstriction. None of this talk is going to really take away from that message, but I've always been interested in the fact that there's an uncoupling of brain blood flow to the cardiac output response. And we see that pretty clearly here with the rapid and large linear increase in cardiac output. And it looks like such a diverse response or a blunted response in the brain. So when I went to Perth, I kind of thought, given Danny and Howard's work in the water exercise, that I might use some water-based exercise to try and investigate the importance of cardiac output on cerebral blood flow during exercise. But as it does happen with Danny's lab, he had a couple other ideas, and he introduced me some clinical folk who presented us with a very unique opportunity, and that is to measure brain blood flow in individuals who've been implanted with a left ventricular assist device, or otherwise known as VADs. So VADs are an established treatment for heart failure, and what they do is by implanting a pump that directs blood flow from the left ventricle into almost what's called an Archimedes screw, and then delivers blood flow into the aorta, they're able to reestablish blood flow and allow for recovery of the heart in congestive or in heart failure. VADs are used as a bridge to transplant, a bridge to explant, and then as a destination therapy. Since 2006, implants have been progressively increasing, and since 2013, approximately 2,500 implants occur each year. They have comparable survival rates to heart transplants, and the primary cause of death in an individual who's been implanted with a VAD is a neurological event or an organ failure. When we started looking at this information, obviously one of the first places we looked was Dr. Patrice Broussard's work, who first investigated brain blood flow during exercise in the middle cerebral artery. And what he was looking at was trying to understand whether or not blood flow responses could be improved in the VAD population. So in this group, this is a continuous flow device, which has a set or a standardized revolutions per minute, which tries to optimize blood flow from the left ventricle without causing any ventricular suction, which would be obviously fairly bad. And he demonstrated that middle cerebral artery velocity does not increase. We don't get that biphasic vasodilation response, and then at maximal exercise, we get a reduction in baseline flow. But if they were able to turn up the pump speed during exercise, and considering that a larger volume was getting returned to the heart, this would allow for more flow to be pumped out. they were able to demonstrate that this biphasic response that we're all familiar with. So we wanted to try and kind of go one step further and maybe look at regional differences, specifically in the middle cerebral and posterior cerebral artery, and then compare the LVAD response to healthy age-matched and sex-matched controls. And you can see in the middle cerebral and posterior cerebral artery, in our healthy control groups, we get that normal submaximal response. We go from baseline to 60% of exercise intensity. But in that LVAD population, not a lot of velocity changes occur. Furthermore, we wanted to compare it to age and sex-matched heart failure groups. And the interesting thing that we see here is that the implanting of the VAD actually elevates resting blood flow compared to the heart failure group. So despite the fact that they don't have an increase in blood flow, they still seem to manage somewhat a higher perfusion than the heart failure group. We also investigated blood flow in the internal carotid artery. And so remember, this takes into consideration velocity and diameter changes. Again, similar responses in our control group as expected. Not a lot of change in the LVAD response to exercise, but again, a similar blood flow at rest. So it seems to reestablish those normative values, which is a greatly improved internal carotid artery blood flow compared to the heart failure group. Given that a lot of the work that we've been doing the last 10 years is related to shear stress, we also wanted to try and quantify how shear rate might be impacting this response. In the LVAD population, that increased or reestablished blood flow elevated shear rate compared to heart failure at rest. However, during exercise, no increase was observed. Whereas when you look at that control group, we see a moderate increase in shear rate. When we think of that hemodynamic response, or we want to investigate what impact that had on brain blood flow function during exercise, in our control group, a 15 to 20% increase in shear rate resulted in a significant vasodilation of the internal carotid artery that was not present in our heart failure group, and did not have, given that the increase in shear rate was not observed significantly in the LVAD population, they actually had a vasoconstriction. So to quickly summarize that first study where we were trying to expand on Dr. Broussard's work, left ventricular assist devices normalize resting cerebral blood flow in heart failure. However, the cerebral hemodynamic response to exercise is impaired in left ventricular assist devices. Now, a product of these individuals having the ventricular assist devices implanted at Fiona Stanley Hospital was that they had to also engage in an exercise cardiac rehabilitation study. So we wanted to set out and ask the next question was, could we use exercise training to improve cerebrovascular function in these patients? And considering the work by Dr. Louise Naylor, Andrew Marano, Anna Shear, and Nacho Suarez, their earlier work showing that exercise training could improve cardiorespiratory function in these populations, we also believe that there would be a cerebrovascular benefit. So in this study, we asked 11 recently implanted VADs to come back or come to our lab and perform a 3-month cardiac exercise rehabilitation study. They performed 3 one-hour blocks of moderate intensity continuous treadmill exercise. On either side of the continuous steady state exercise, there was a skeletal muscle warm-up, and then on the other side, a skeletal muscle cool down as well. Intensity was based around 50 to 60% of VO2 reserve, and similar to the Preventia study, we progressively increased intensity over the 12-week intervention. All of our individuals came to the lab pre and post intervention and conducted a baseline as well as a recumbent cycling VO2 max test. Our starting workloads were 15 watts, and then we increased that wattage every 2 minutes until exhaustion. We statistically analyzed this using a 2-factor repeated measures ANOVA, and then provided a Bonner-Perroni correction for all the groups and exercise intensity interactions when we observed them. Our instrumentation setup looks like this. This is one of our ventricular assist device subjects. You can see we have our transcranial Doppler, our cardiorespiratory measures, Finopress in the event that we were able to quantify it. However, most of our VADs, we had to use a Doppler approach in the oscillometry method. And then also 12-lead cardiac ECG. During this time, I would stand next to the subject and also measure internal carotid artery flow. And if you can see here, this is the resting ICA velocity profile that we see. We've got diameter here. And again, these VADs most of our subjects had what's called a heartmate 3. And so there was supposed to be some brief shift in the recycling of the pump, which allowed for a more pulsatile response. However, when you look at that response, it doesn't look like a normal pulsatile response as we think of in terms of a systolic and a diastolic response. When we look at the response during exercise pre-training, our individual, we didn't see, again, much change in that velocity profile. However, when we compare that velocity response post-exercise, this is the same subject, you're seeing that that individual has reestablished some of its cardiac functioning, and we're now seeing significant increases in systolic pulsatility. When we group this response and measure the internal carotid artery flow, in our pre-training group during exercise, a small increase at 20%, but not significant, which then resulted in a reduction in blood flow for the remainder of the intensity increases. However, post-training, there is a modest and significant increase in blood flow velocity in the internal carotid artery at 40 and 60%. However, most of this increase in flow is unrelated to a change in diameter. Both groups still had significant reductions in diameter during exercise, which leads us to the conclusion that the increase in velocity post-training, or increase in blood flow post-training, was largely due to increases in velocity in our LVAD trained group. Given the importance we've been placing on shear stress, we wanted to also investigate this. So remember, the diameter has decreased, but velocity has increased. So that will then press a larger increase in shear stress on the endothelium of these individuals. So much larger post-exercise shear stress, but unfortunately, it did not seem to have an impact on diameter. I already mentioned the importance of CO2. So given that it wasn't the improvement in cerebrovascular response wasn't endothelial mediated, we figured we better look at another mechanism. And when we look at CO2 during exercise here on this graph, you see a significantly greater hypercapnic response during the submaximal exercise in our post-training LVAD population. And when we correlated the change in flow with CO2, it was a very modest correlation, but nonetheless significant. So to quickly summarize the findings from this, cardiac exercise training does improve cerebrovascular function in individuals living with ventricular assist devices. And this is really important, given that we're seeing a larger number of these devices being implanted as a destination therapy, not necessarily in Australia, but in the US, we're seeing about 60% of the population or implants being used for a destination. Most of these improvements in cerebrovascular function appear to be related to cardiorespiratory improvements, not necessarily cerebrovascular endothelial dysfunction. And I just want to say, you know, it's been a great pleasure being part of the Green Ainsley Army, and thank Pat and Patrice and Caroline for putting on these talks. And just to kind of some evidence, we obviously don't take ourselves too seriously. You know, this is just another one of these amazing Ainsley Green Research Lab talks. Thank you.
Thank you, Kurt. We have a question from Mike Timko. Says, do you know if the end-tidal to arterial CO2 gradient would be similar or different in the LVAD patient?
I wonder, I'm sure that exists, the data exists out there. I haven't looked at it, but it'd be interesting to consider, given that the blood flow through that pulmonary circulation isn't really going to change too drastically, is going to change drastically following the VAD implantation. But unfortunately, I don't have that information. But a great question, especially considering that small CO2 change that we saw pre and post training.
We have another question from, how did blood pressure change in the VAD group, particularly pre versus post-exercise? How much does this contribute to the cardiac output and cerebral blood flow associations described?
Yeah, and apologies, I didn't show any of the blood pressure response. So the BP response at the same relative intensity was a little greater, but that's primarily due to the fact that these individuals had improved their fitness. So they were exercising at higher wattages for the same given intensity. In terms of the cardiac output, unfortunately, we didn't have any of those measures in this population, but it's still something that maybe we could look at. I think the biggest limitation is that we didn't have a continuous measure of blood pressure in that group, and it would have been. I was really hoping that the Finepress was going to reliably allow us to do that, but we weren't quite able to. And then I think the next question would be is trying to do that while also having a measure of cardiac output. Unfortunately, I'm sorry, I can't really answer that question any further.
Well, I have a question for you, Kurt. So you showed differences in shear stress between the groups, or pre and post exercise training, sorry, but you also showed quite different blood flow patterns. What do you think is the relative importance of, the mean shear stress that you're getting over an average cardiac cycle and the actual like velocity waveform that's contributing to, peak and diastolic shear stress? Is reestablishment, of that pulsatile waveform potentially more important than increasing mean shear, or do you think it's the increasing mean shear that's the most important?
That's a good question. I think the one thing that we want to remember is that the reestablishment of the pulsatility only occurred during exercise. So we still needed the heart to become loaded again, or have enough volume in it to get that, to have that pulsatile response. I am interested in how that mean shear profile impacts endothelial function. And we didn't really, I wasn't present when we assessed it. However, Andrew Meirano and them and Danny's group have also measured the flow-mediated dilation response in those individuals. But I don't quite have that data on hand, but I believe there was an improvement in FMD following the training in this population.
Very cool. And then we have one more question from Carolyn Ricards, and then we'll move on. Excellent talk, Kurt. Do you know if cerebrovascular responses differ between people with pulsatile flow LVADs versus continuous flow LVADs? I'm curious if the chronic pulsatility of flow modifies cerebrovascular function and adaptations to exercise in the long term.
I don't really have a response to the long-term implications of whether or not pulsatile versus non or continuous flow pump impacts cerebrovascular function. I think the first study we showed, some of those individuals had been living with VADs for a longer period of time, and they still never really had an improved left ventricular function, or cerebrovascular function following LVAD implantation. So I think without engaging in exercise capacity and having some form of reestablishment of that or recovery of that cardiac muscle, I'm not certain that we see much improvement other than the initial improvement that we see, or that demonstrated where we increase mean flow. The other thing that's probably important to look at is that the pulsatility that's induced by the HeartMate 3. I mean, at this point in time, I haven't seen any other data come through, but it still doesn't look like a normal pulsatile response. So I think there's some really interesting things we could do. And while it's not possible to have a VAD patient get an MRI, I'd love to see what this profile looks like if we modeled it through some of the work that Hannah and Harrison were presenting.
Awesome, thank you. We'll sneak one more in really quick because it's from the LVAD OG, Dr. Patrice Broussard. Should LVAD pump speed be manipulated during cardiac rehab to optimize cerebrovascular responses during exercise in these patients? I feel like he might be biased in his opinion.
I think it'd be, I mean, you know, I think the golden age will be when these pumps are, you know, are connected to some form of smart device that does this for us automatically. We did talk about this, was how could we come up with the right mode of optimizing or enhancing cerebral blood flow in these populations, or in this population, similar to what Danny and Howard were hoping to do with the water immersion. Like, could we provide a situation where we increase the blood flow response, but we couldn't quite get ethics to allow us to go in and out and tamper with the pump speed like Pat and the Danes were able to do.
Fair enough. Awesome. Thank you for your talk, Kurt.
Thanks, guys.
I think, Danny, are you wanting to comment?
Well, if I'm allowed to, I'm not sure if that's against the rules, but I'll do it anyway.
We'll get Hannah to set up. Will you?
Oh, terrific. Because I'm really engaged with this series of questions, both, I mean, I think Patrice is right onto it. Clinicians generally optimize VAD outputs based on an echo done at rest. And that's partly not to involute the ventricle and make sure they've got the settings optimized, but then they don't take into account the fact that people are getting a VAD to do some exercise and improve their peripheral skeletal muscle. So you're right on the money, Patrice, they're measuring the wrong outcome. And, you know, for the engineers in the audience, there's a possibility, an opportunity here to have VADs that are somehow detecting what's important, which is brain perfusion. I mean, the entire cardiovascular system is based around maintaining it. So why shouldn't VADs be? So you're right on that, Patrice. And as for the questions that came, I think from Caroline, great question about the pattern of blood flow. I mean, I can't help myself because we did a decade of work on different patterns of shear stress, Dick Tyson and I, trying to work out whether the pattern of shear made a difference compared to mean shear. And the brief answer to that decade of work is yes, it does. And that anterograde shear is good and retrograde shear is not so good. And VADs have really aberrant shear. You know, James Fisher asked whether the blood pressures change. Well, you can't even measure blood pressure in these guys in a conventional sense because they don't have a pulse. So, yeah, it's a very interesting model for those of you who have access to it and are interested in this area. There's a lot of interesting work to do where there's a natural experiment happening here, or a clinical and natural experiment. That's not an oxymoron. Thank you.
Great. Thank you for that, Danny. So up next, we have Hannah Caldwell. So Hannah Caldwell is a PhD student at the University of British Columbia, Okanagan with Phil Ainsley. And aside from this talk, I would recommend keeping an eye out for some of her emerging work. I know over the last couple of years, she's done some very interesting work looking at the mechanisms of cerebrovascular function as it pertains to CO2 and pH, which I think we would all be very interested, especially after the theme of today's talks. But today she's going to present some work from the UBC UWA collaboration. You may recall that Dr. Green highlighted some peripheral blood flow work where they looked at removing the shear stimulus from an exercise or a heat blood flow response. Hannah's going to present some work kind of looking at this through a different lens and what the implications might be for cerebrovascular function. And so the title of her talk today is The Influence of Sustained Elevations in Cerebrovascular Shear Stress by Hypercapnia and Exercise on Neurovascular Coupling.
Great, thanks so much, Ryan, for the introduction. And thank you to Caroline and Patrice for organizing this excellent seminar series. I'm really excited to talk today, and there's been some pretty incredible talks so far. So during exercise, cerebral blood flow increases by approximately 10 to 20% progressively, with increases in exercise intensity, up to approximately 70% of maximal oxygen uptake, or VO2 max, to regulate cerebral substrate delivery. And this is mediated via increases in cerebral metabolism, as well as alterations in arterial carbon dioxide. So during exercise, we have an increase in the volume of expired carbon dioxide, as well as a lesser relative increase in alveolar ventilation. And as such, we have an increase in the partial pressure of arterial CO2. And these changes in CO2 regulate cerebral blood flow such that at approximately 60 to 70% of VO2 max, we have a hyperventilatory response that reduces CO2 as well as cerebral blood flow back towards resting values. The cerebral vasculature is exceptionally sensitive to CO2, such that within the arterial circulation, we have an increase in arterial diameter with increases in carbon dioxide as well as reductions in perivascular pH. The sensitivity to CO2 exists throughout the cerebrovascular tree, including the intracranial cerebral circulation. And here we can see changes in middle cerebral artery diameter with alterations in carbon dioxide. And further, the extracranial arteries are also vasoactive, where here we can see an approximately 20% change in arterial diameter across a wide range of carbon dioxide. Increases in cerebral blood flow elicit shear stress within the artery, releasing vasodilatory signals such as endothelial nitric oxide synthase activation. And this release of nitric oxide acts on the vascular smooth muscle to elicit vasodilation or an increase in arterial diameter. As well, hypercapnia increases cerebral blood flow and shear stress and provides a model to experimentally match the cerebrovascular response of exercise. Neurovascular coupling describes the relationship between metabolism and blood flow in the human cerebral circulation, where we have increases in cerebral metabolism that lead to a localized vasodilatory response to increase cerebral blood flow in response to a matched increase in metabolism. And we measure neurovascular coupling using a standardized eyes open, eyes closed visual stimulus, whereby we measure posterior cerebral artery blood velocity, supplying the visual cortex in response to a visual stimulus. So we complete 5 repeated cycles with a standardized visual stimulus such as a flashing checkerboard, and the hemodynamic response to these 5 cycles is averaged across and presented as a mean value for each individual. We extract this data on a beat by beat and breath by breath basis to temporally adjust for spontaneous changes in blood pressure and partial pressure of arterial carbon dioxide, as well using a cubic spline interpolation with a custom designed MATLAB code. And importantly, neurovascular coupling is a repeatable and robust technique that is reportedly impaired in certain clinical groups, such as stroke, hypertension, and diabetes. So why do we think CO2, or in other words, shear stress, would affect neurovascular coupling? We know that the peak initial increase in cerebral blood flow within this neurovascular coupling response represents neural feed-forward glutamatergic signaling, and this includes nitric oxide synthase activation. And recently we've shown that approximately 30% of this peak initial increase in cerebral blood flow is responsible by nitric oxide mediated pathways. So therefore, the pathways involved in neurovascular coupling are upregulated with shear stress via nitric oxide signaling. The aim of this study was to assess the influence of matched increases in cerebral artery shear stress induced via hypercapnia and exercise on neurovascular coupling. And we hypothesized that matched increases in cerebral artery shear stress induced via hypercapnia and exercise would both improve neurovascular coupling. So the main results from this trial have been previously published by Kurt Smith, Ryan Hoyland, Danny Green, and co-authors, where they showed matched increases in cerebral artery shear stress, irrespective of stimulus, induced similar changes in extracranial artery diameter in humans. And for the present data, I'm going to present a pilot sample of 10 healthy males, approximately 25 years old, who completed 3 randomized counterbalanced interventions, approximately 30 minutes each. So participants completed hypercapnia, so 4.5% inspired carbon dioxide, submaximal cycling at approximately 60% of heart rate reserve, as well as a resting time match control. And through pilot testing, these levels of hypercapnia and submaximal cycling were determined to be able to elicit a robust increase of approximately 30% in shear stress, which allowed us to randomize and counterbalance the order of these two conditions. So as previously reported, there was approximately 20 to 30% increases in both the internal carotid artery and vertebral artery shear stress that was matched during both 30 minutes of exercise and hypercapnia. And additionally, intracranial cerebral blood velocity was measured continuously in real time, and levels of inspired CO2, as well as the exercise intensity, were adjusted accordingly to ensure an appropriate matched increase in both of these shear stress stimuluses. So here we have cerebral blood velocity on the y-axis and time across the x. And as you can see throughout the control condition, there was no change in cerebral blood velocity. Whereas in the CO2 condition, there was approximately 30% increase in cerebral blood velocity that was well matched during the exercise trial. So we were able to successfully increase shear stress to the same extent during both hypercapnia and exercise. So for our primary results, again, looking at the posterior cerebral artery, which supplies the visual cortex during a visual stimulus. First, we'll look at, prior to each intervention, the neurovascular coupling response. So on the y-axis, we have the posterior cerebral artery cerebrovascular conductance and time across the x-axis. The data presented here, the mean lines represent the group means in the solid lines, as well as the respective standard deviation in dashed lines. And again, these are cycle averaged across 5 cycles for each individual. At time 0, you can see participants open their eyes and there was a robust increase in posterior cerebral artery blood velocity that was importantly not different prior to each of the 3 interventions. Following each of these trials, same, we have the posterior cerebral artery cerebrovascular conductance on the y-axis and time across the x. And within this sample size of 10 participants, there was no significant difference between this neurovascular coupling response following each of these 3 trials. However, we have a few ideas on how we can further follow this up and explore the relationship between shear stress and neurovascular coupling to elucidate whether there is an effect of shear stress. So just to recap what we currently know about exercise and neurovascular coupling, we know that the NVC response is not altered during submaximal exercise at approximately 50 to 60% VO2 max, and this is with or without controlling for the partial pressure of CO2. So in 2011, Chris Willey and colleagues showed that during submaximal exercise, when participants were poikilocapnic or breathing uncontrolled, and CO2 was allowed to fluctuate during exercise. As you can see, although cerebral blood flow is higher during exercise, the absolute response in this visual stimulus, or the absolute neurovascular coupling response, is not different during exercise than versus rest. And we followed this up recently, again, during submaximal exercise, but here participants were under isocapnic conditions where we controlled CO2 during exercise and matched to levels at rest, and again showed no effect of neurovascular coupling during exercise. So for a brief discussion on these findings, sustained shear stress via CO2 did not improve neurovascular coupling, and additionally exercise did not improve NVC. But in terms of future directions, we know that blood pressure, partial pressure of CO2, and metabolism may be influencing the neurovascular coupling response following exercise and hypercapnia within the first hour. So we'd like to explore this further with respect to the time course of these changes following exercise and hypercapnia, as the current study reported neurovascular coupling approximately 20 minutes following each of these acute interventions. And additionally, we'd like to investigate the effect of repeated visits with an acute exercise or hypercapnia shear stress training to see if there's an effect of repeated episodic bouts of shear stress, either induced via exercise or CO2, on the neurovascular coupling response. Lastly, I'd just like to thank my supervisor, Phil Ainsley, as well as my lab mates and our funding organizers. Thank you.
Great talk. Thank you, Hannah. So we have one question already from Tracy Baynard. Fabulous presentation. Do you believe you'd have similar findings using a different type of neurovascular coupling stimulus, say some type of psych test, for example, the flanker, Stroop, or others? The question being related to different regions of the brain being activated. This may be of interest in relation to Alzheimer's disease or dementia in conjunction with exercise.
Great, so thank you for the question, Tracy. That's an excellent question. So in our lab, we've previously used the approach of the visual stimulus and measuring blood supply to the posterior circulation, as we found that that is the most repeatable and robust stimulus that we can elicit each time, irrespective of whether we were measuring blood flow, for example, in the anterior circulation in response to a cognitive test. We find that although that may be a little bit more practical and clinically relevant for groups like Alzheimer's disease or dementia, specifically when we're testing young, healthy individuals, we find that the visual stimulus is the most robust measurement for neurovascular coupling and the most repeatable. But I think it'd be definitely interesting whether we were testing clinical groups to try a more relevant test that might be able to assess some sort of regional specificity.
Awesome, great answer, Hannah. While we wait for a couple more questions to hopefully filter in, I'll ask you something. So one of the things that has definitely been a reoccurring theme across all of the presentations today has been how shear stress is being manipulated and the nuances of how we might manipulate shear stress to elicit the best effects, because I think the majority of this work is looking at how can we use shear stress as a tool to improve cardiovascular health and specifically cerebrovascular health. And so maybe just touch on a little bit more how you're able to match the shear stimulus with exercise and with CO2.
That's a great question, thank you. And I think probably the most novel and exciting aspect of this study was the experimental design. So the ability to match the shear stimulus during submaximal exercise as well as with hypercapnia and how we did that. So as I said, through pilot testing, we were able to determine approximately the range of exercise intensity and hypercapnia that we wanted to achieve. So that 4.5% of inspired CO2 was titrated in real time, approximately between 3 to 6% inspired CO2, depending on the shear stress that we wanted to elicit. And that's the same for the exercise intensity. There's approximately 60% of heart rate reserve, but that range between 100 and 120 beats per minute heart rate. And kind of our main metric of shear stress during either intervention was continuously measuring the middle cerebral artery blood velocity using transcranial Doppler and being able to elicit a 30% increase that we were able to watch in real time and titrate those levels of CO2 and exercise intensity accordingly to really make sure we hit that target.
Awesome, thank you. And then maybe one more question before we move on. So there was no difference in the CO2 and the exercise and control responses following each intervention, but it definitely looked like the lines started to diverge, and you talked about some reasons why that may have been, but is there anyone that you can kind of point to as being the most important? Because you did present conductance values, and so it might not necessarily be blood pressure, or perhaps the relationship of blood flow and blood pressure is changing, but just maybe if you could shed some more light on that.
Yeah, absolutely. So, so obviously cerebral blood flow being multifactorial and incredibly integrated in its regulation, I think it's definitely difficult to discern the blood pressure response. Although I presented the cerebrovascular conductance, we typically do that in order to also account for temporal changes in the NVC response, particularly when we elicit the test. Some individuals will exhibit a pressor response or an increase in blood pressure. So we're able to correct for those changes. But whether the actual baseline blood pressure and any prevailing changes following either intervention is affecting our NVC response, I think that's what we're most interested in following up. as I said, sort of a time course following exercise within that first hour. Because I think, for example, if blood pressure were slightly lower during exercise or slightly higher following hypercapnia, that's going to influence that NVC response depending when we take that post measure. And then I think kind of the other really interesting factor that pretty difficult to investigate, but whether we're actually seeing different changes in cerebral metabolism during either of those interventions, and if that's going to contribute to some resting change that would elicit a differentiation between our NVC response. I think that's pretty hard to tease out, but definitely could be a candidate for why we're seeing differences.
Awesome, thank you. So great talk and great answers to the question. So up next we have Jay Carr, who I'll introduce while he gets his slides up. We finally made it. This is the last talk of the session for those who are watching in their respective evenings. So Jay Carr is a PhD student in Phil Ainsley's lab at UBC Okanagan as well. And since arriving at the lab at UBCO, Jay has immersed himself into this area of cerebrovascular shear stress and cerebral blood flow function, and has some very interesting ideas moving forward. And while the talks thus far have looked into where the functional cerebral blood flow responses, and how these may or may not change following different types of shear stimuli, what Jay is going to touch on today is about what exact responses maybe do we want to measure after shear stress, and what do the responses we measure after any sort of training regimen potentially mean. And so with that, the title of his talk is Cerebral and Peripheral Vasodilator Function in Young Healthy Humans. Go ahead, Jay.
Yeah, I'm conscious that I'm following 7 phenomenal talks and the audience might be quite frazzled, so I'm just going to get stuck straight into it. The focus of the study that I'm going to present was to investigate the relationships between cerebral and peripheral responses to transient increases in shear stress. As described by Danny earlier, we know that peripheral arteries adapt with regular exercise, and we know that shear stress is one of the major components promoting this vascular remodeling. We test this in vivo in the peripheral vasculature through flow-mediated dilation, and that is to say large artery reactivity to shear stress. High flow or stress dilates arteries, and low flow or shear stress constricts arteries. And this is a largely nitric oxide mediated and endothelium dependent mechanism. By assessing FMD in vivo, we gain insight into the functionality of the endothelium and the health of the vessel in question. And because FMD and coronary artery function have been significantly correlated, we also gain some insight into cardiac health. And FMD is generally predictive of overall cardiovascular health. And so improving FMD equates to decreasing the risk of cardiovascular events. Because of this, the test FMD is used to assess vascular adaptations of exercise and the impairments of disease. The cerebral benefits of exercise are becoming apparent, as has been discussed at length so far, but in vivo, the vascular adaptation aspect of this remains somewhat less understood than it is in the peripheral vessels. While many tests tell us about the functional activity of the brain's vasculature, reactivity to carbon dioxide, or various blockades, neurovascular coupling, etc., many of these responses represent several overlapping mechanisms. As Howard put it earlier, when we test the cerebrovasculature, we tend to just throw the kitchen sink at it. In regards to today's theme of shear stress, none of these tests specifically assess endothelium shear-mediated function in the large cerebral arteries. So if we want to test the effects of exercise or the impairments of disease on the endothelium in vivo in the brain, we need an in vivo assay which specifically assesses cerebral endothelial function. In the early 90s, it was shown in animals that cerebral arteries are also responsive to shear stress, and later work by many of today's presenters showed similar responses in humans during exercise in both with and without hypercapnia. This was developed into the cerebral FMD mentioned a little earlier. Analogous to the peripheral FMD, cerebral FMD evokes dilation in the cerebral arteries through transiently increased shear stress. And while this is yet to be published, our lab, along with, under Ryan Hoyland and others, have carried out CFMD with and without nitric oxide blockade and found that it was 40% reduced after LNMMA, showing that CFMD is at least partially NO mediated. In the same study, ICA dilation in response to a standard CO2 reactivity test was unchanged, showing that it's perhaps not NO mediated. And I say perhaps because while we show here that CVR is unaffected by NO blockade, previous studies have found that NO plays a somewhat permissive rather than obligatory or necessary role. Nonetheless, endothelial nitric oxide is necessary for cerebral FMD, and so it's a test of endothelial function. So our primary outcome was to compare the magnitudes and assess relationships between endothelium-dependent dilation in the brachial artery and internal carotid artery. And we also aimed to determine the relationship between shear-mediated function and a common test of cerebrovascular function, that is to say, CO2 reactivity. And finally, in order to determine whether differences or similarities between the arteries could be explained by differences in vascular smooth muscle function, we also compared endothelium-independent function. In 19 healthy young participants, we used end-tidal forcing to control end-tidal CO2 and O2 breath by breath, duplex ultrasound to continuously assess the brachial artery and internal carotid arteries during the relevant tests, phenometer for continuous arterial pressure, and ECG for heart rate. To assess endothelium-dependent function, we used CFMD of the internal carotid artery and a standard brachial artery FMD. The CFMD test consists of a 2-minute baseline, 30 seconds of isotoxic hypercapnia at 9 millimeters of mercury above baseline, followed by a further 3 minutes of scanning to capture peak shear and diameter. Brachial FMD consists of a 1-minute baseline, 5 minutes of arterial occlusion at 220 millimeters of mercury or above. And again, this is followed by a further 3 minutes of scanning. We also compared CFMD against CO2 reactivity of the internal carotid artery, and that is to say the blood flow response during a single 5-minute stage of hypercapnia at 9 millimeters of mercury above baseline. And finally, to assess endothelium-independent function, we administered 400 micrograms of glyceryl trinitrate, an exogenous nitric oxide donor, and scanned both arteries for the following 10 minutes. So all tests had the desired dilator results. Here you can see diameter percent change from baseline on both y-axes over time on both x-axes. And these figures are mean and standard deviation of all the individual data in 5-second bins. There were transient elevations in shear stress, which led to transient vasodilation in both the internal carotid and brachial arteries. On the left, again, we have percent change in diameter over time on the x-axis, showing that the GTN significantly dilated both arteries. On the right, we have relative blood flow reactivity of the internal carotid artery, or relative velocity reactivity of the middle cerebral artery. Steady state CO2 caused the expected increases in mean blood flow and mean velocity. Additionally, mean arterial pressure and heart rate were also significantly elevated during reactivity. However, the main findings were that between arteries, endothelium-dependent responses were not significantly correlated, nor were the endothelium-independent responses. And finally, CFMD diameter change and CO2 reactivity of the internal carotid artery were also not correlated. So for our main outcome, comparing and assessing the relationship of endothelium-dependent function between the arteries, we found that FMD is not reflective of CFMD and vice versa. And a look at the hemodynamics of both arteries can give us some insight as to why. Arterial shear-mediated dilation is influenced by both baseline diameter of the artery and shear stress stimulus. But the degree to which changes in shear stress modulate vessel diameter is also determined by numerous other hemodynamic characteristics. For example, baseline shear stress markedly differs between the arteries. Resting shear in the ICA is quite high, whereas resting shear in the brachial artery is comparatively low. And conversely, the hyperemic responses in the ICA amount to shear increases of about 20%. Brachial artery shear increases by about 1,000%. Crucially, hemodynamic conditions determine endothelial cell phenotype. Endothelial cells are most differentiated in the microvasculatures of organs, as can be seen in these figures. where they have distinct characteristics that allow for organ-specific functions. And while brachial and internal carotid endothelial cells aren't as phenotypically distinct as, say, the blood-brain barrier compared to those in the liver, for instance, nonetheless, there is differentiation between large arteries where hemodynamic conditions are consistently different. As such, there's regional heterogeneity in regards to the predominant mechanisms of shear stress transduction and sensitivity, as well as differentiation of many other factors due to differences in arterial topology. The brachial artery, for example, is relatively straight with mostly laminar shear, whereas the internal carotid artery is exposed to more turbulent and disturbed flow as a result of proximity to the carotid bifurcation. And this results in differences in the production of reactive oxygen species, nitric oxide, inflammatory and adhesion molecules. And as you can see, there's considerable between variation in carotid bifurcation geometry as well, which no doubt contributes to the variability of downstream endothelial characteristics and to the lack of correlations between endothelium-dependent function of the ICA and BA. Recall from Hannah and Harrison's talk, even twins have huge differences in vessel structure and function. So given regional endothelial heterogeneity, as well as other differences between regions, in order to determine cerebral endothelial cell dysfunction, that region should be tested directly with an assessment which narrows down the potentially involved pathways. By no means are we trying to imply that FMD and CO2 reactivity tests don't have their place in both clinical and research settings. Of course they do, they're very, very useful. FMD is definitely associated with adverse cardiovascular outcomes, as has been shown in several studies since the 90s. And impaired CO2 reactivity is associated with all-cause mortality, as well as with Alzheimer's disease, and possibly with stroke risk, although this has only been shown in patients with existing carotid stenosis, as far as I'm aware. But the point here is that neither of these tests tell us specifically about the cerebral endothelium or endothelial nitric oxide mediated pathways, and whether it's these which are in decline in these diseases. So we suggest that although CVR assesses vascular responses in the brain, it's a marker of general vascular function rather than a specific test of any one mechanism or pathway. The development of novel targeted therapies that are vessel bed specific is prefaced on the specificity of research and clinical tests and assays. As such, in future studies assessing brain arterial function, if large cerebral artery endothelium is the primary outcome, for the assessment of either exercise-induced adaptations or early detection of impairment, as Danny was discussing earlier, then a combination of these tests would give us more information than one of them alone. In order to further investigate the prognostic value of these tests, specifically CFMD, over the next couple of years, we're also going to assess clinical populations with varying degrees of carotid stenosis, both before and sort of after carotid endarterectomy. Or those with carotid webs or atherosclerotic burden, as well as in diseases affecting the cerebral microvasculature. The take home here, though, is that just because someone has a highly responsive peripheral endothelial function or cerebrovascular reactivity to CO2 doesn't necessarily mean that they'll have an equivalently great cerebral endothelial function, and vice versa. And this is because neither of the former two tests examines the cerebral endothelium specifically. Thank you very much.
Awesome, thank you for that. Jay, we'll get on to the questions here. So the first one from Caroline Ricards. Thanks for the great talk, Jay. You tested healthy subjects in this study. Regarding clinical populations with impaired vascular function, can you speculate on whether responses would be more aligned between vascular beds or even more different?
I mean, there's certainly relations between peripheral FMD and CVR. As I was saying, I think CVR is a pretty good indicator of cumulative vascular damage. As for whether the relationships that I showed earlier would be stronger in those with vascular dysfunction, I can't yet say. That's why we're gonna do those studies. I think there's such huge variation in internal carotid and common carotid shear stress patterns that I honestly don't know if the relation would grow stronger with overall vascular dysfunction, or if it would just be a similar lack of correlation. Possibly stronger, yeah, maybe. Was my video off the whole time? I had no idea.
Nice, thanks for that, Jay. So another question from AJ Rosenberg. Thank you for a great talk, Jay. I have a two-part question. One, what was the rationale for the 2 minutes hyperventilation, 20 breaths per minute prior to the hypercapnia stimulus if you clamped at baseline entitled values? And then two, have you compared the shear stress and ICA dilation responses with and without hyperventilation prior to the CO2 stimulus?
Okay, I'll tackle those one at a time, because by the time I've answered the first one, I've probably forgotten the second one. The first one was why did we hyperventilate during this CFMD test? Sort of a couple of reasons. During the sort of rapid elevation in end-tidal CO2, even though we're using end-tidal forcing and it's pretty rapid as it is, if people were breathing at their normal respiratory rate, getting them to +9 above baseline would probably take around about 30 seconds, if not longer, sort of a few breaths. But if we have them hyperventilate, then we can get them to, +9 in a matter of sort of 10 seconds or thereabouts, or even less. And so that's pretty much the main reason. Additionally, though, it makes it slightly easier on the sonographer if there isn't a sudden jump in ventilatory drive with the onset of CO2. So if the participant is already breathing relatively fast, then the sonographer's got, got to adjust that image or adjust that, position or comfort 2 minutes prior to the actual test, or we just wait until they get the image sort of dialed in before we raise PET CO2. And then, yeah, as I said, I've forgotten the second part of this question already. Could you remind me, Ryan?
Yeah, so part two is, have you compared the shear stress and ICA dilation responses with and without hyperventilation prior to CO2 stimulus?
I don't know that we have, but Ryan, maybe you could speak to that.
Yeah, so this isn't something that we have addressed specifically. I think the one important thing to note is that when we get the individuals to hyperventilate prior to this test, even though they are breathing more, we're keeping their carbon dioxide the same. And so, there's some evidence that prior hyperventilation before a CO2 reactivity test might impact the magnitude of the response we see, but that's in the context of the hyperventilation occurring with simultaneous hypocapnia. And so we haven't looked at that, but we wouldn't necessarily expect it to lead to any changes. One of the reasons, I think this plays into it to touch on some of the things Jay mentioned, is with the hyperventilation is the changes in diameter we're detecting are quite small. And so one of, I guess, the design goals for that was to keep everything as controlled as possible. And so large changes in ventilation could cause large changes in neck muscle movement, which would make it much more difficult to detect changes in diameter, or it might lead us to detect erroneous changes in diameter. And so it's more methodological than necessarily physiological reasons why that's in place for the test, but that's a good question. So moving on, we have another question from Justin Sprick. Very nice talk. Thank you. Do you think the regional heterogeneity would still persist in patient populations that are characterized by an elevated resting peripheral and cerebrovascular flow at rest, such as anemia, in which flow is increased due to both a decreased viscosity and a reduced oxygen delivery?
Great question. Yeah, I think it would. If anything, regional heterogeneity in the factors that I mentioned would probably be more pronounced. Maybe. In regards to Hannah and Harrison's talk, I think was really, really interesting. Just the way that there can be such divergence of intracranial cerebral artery geometry. You know, they didn't show the internal carotid artery there, but that's been shown previously. It can be huge, and it only gets worse as you get older. And so I can imagine that would probably get worse in pathological situations as well. So yeah, I imagine heterogeneity would be increased and probably worse. As for whether or not it would be, would have recourse to, you know, viscosity specifically or low oxygen delivery specifically, I'm not sure. Viscosity would, yeah, definitely play into it. I'm not so much sure about oxygen delivery though.
All right, we have two more quick questions. I think one is more of a comment, and I think this gets at the fact that we've had a lot of interesting questions already, but from Tracy Baynard, great talk, a lot of food for thought, and ties in nicely with Laughlin's work along the arterial tree. It will be interesting to see data collected in due time that might help characterize different endothelial dysfunction phenotypes. And what I think that this really highlights is that the data you presented today, Jay, is, well, I think it provides some answers. It definitely provides more questions than answers, and is really a starting point for several other lines of investigation that I know you've already looked into and have planned. And then the final question is from Danny Green. Brachial, radial, and coronary arteries are more or less similar in size. Brachial as a surrogate for coronaries makes some sense, but carotids are large and hence less reactive. A good surrogate for small intracerebral artery health? That's a good question.
Good comment, question, Danny. I couldn't agree more. If sort of the direction is like, you know, should we be looking at internal carotid artery endothelium dependent function as a surrogate, as you say, for intracerebral artery health? No, perhaps we shouldn't be, because as I say, you know, the degree of endothelial heterogeneity is between, you know, ICA and the smallest PL arterioles is probably only going to, you know, be even greater. But what I think would be really interesting is the relation between ICA dysfunctional function and carotid bifurcation and bulb dysfunction, where, you know, carotid atherosclerosis is prominent. I think that would be super interesting. I think, yeah, so in my, you know, discussion slide there where I mentioned future directions, the next and most important future direction for these studies for me is the carotid endarterectomy and stroke populations. The future studies involving, you know, microvascular dysfunction, they're super interesting to me, and I'm really excited to go down that route. But I think it's a little bit less likely, or a little bit more far-fetched to start expecting great and obvious relationships there.
Jay, I've unmuted. Can I jump in? Is that okay, Ryan?
Yeah, go for it.
Sorry to do that, Jay. That's a great answer. I think you've nailed it. That like for like surrogate for carotid disease is a fair answer. We do have a problem with the amplitude of the carotid responses compared to brachials or radials, where you've got a larger signal in the FMD response than you do in a carotid. But the other thing it makes me think about is the old FMD literature, which, and I'll draw your, for those of you unfamiliar, I'll draw your attention to the FATE study performed in Canada on firefighters and their endothelium, which showed that there was predictive capacity in the blood flow response or down the brachial artery just as much as there was in the brachial artery diameter response, and that those two things may predict different types of diseases. So it might be that, you know, as a surrogate for microvascular disease in the brain, we might end up looking at other measures rather than diameter responses in the extracardials. So don't throw the baby out with the bathwater. This is all very early days, and I'm sure we'll find, but it's going to take some longitudinal studies to tease all that out, and they're very hard to do. Thanks. That's all.
Yeah, that's a really good point, Danny, for sure. I think the response dynamics and characteristics of all of the tests that I mentioned are super important and really interesting as well. It's not just percent change of diameter, although that's what we've been talking about the most. Yeah, absolutely. I think that's probably going to play into future studies as well. And the CFMD test, you know, you guys pioneered it. So it's been underway for a few years now, but it's still sort of in its relative infancy in the grand scheme of things. And so I think there's definitely development to be had. And yeah, I think the reactive hyperemia aspect of a transient response to CO2 is super interesting as well.
Awesome. Thank you, Danny, for that question, and Jay for your insightful responses. I think we'll wrap up there. If we were in Australia, we could probably head to the pub to discuss more, but unfortunately, I don't think that's the case for many of us. So I'll just pass it back to Patrice Broussard for a second here.
It's still a bit early here for that, Ryan, unless Phil Ainsley is in town. That makes all the difference.
So thank you to Bara, Kurt, and Ryan for the organization of such a high-level seminar, and of course thank you to the amazing speakers involved in that seminar. And make sure to register for our next seminar, which will be held on October 28th, and it will be chaired by Dr. James Fisher, and it will be about cerebrovascular dysfunction in disease states, and his talk will be about the cerebrovascular dysfunction in atrial fibrillation, with the abstract talk from Mathilde Paré, Zoe Adams, and Mickey Fan. So again, thank you for your attendance, and see you soon.
Thanks so much, everyone.